I'd like to welcome everyone. Today we have the pleasure of having Dr. Alessandro Alessandrini on our last lecture of the series. In this series, we had a mean of, on the past lecture, we had about 53 people that joined it, which was amazing. And we'd like to thank you, all of you, for joining, being active in asking questions and participating with your group. Again, we would like to make this as the most interactive possible. Please use the Q&A on the bottom for ask your questions or raise your hand by clicking on that button on the bottom of your Zoom. And if you have any problems, you know how kind of to chat with us, either myself or Thiago. And at the end of the lecture, like we did before, we'll get you an email so we can continue to improve this series and get further feedback. If you have suggestions of other topics and things that we should include, also please let us know. I'll pass it on to Thiago to do a formal introduction of our speaker today. Thank you.
Thanks, Leo. So today is my pleasure to introduce you Dr. Alessandro Alessandrini as our speaker. Dr. Alessandrini is Assistant Professor of Surgery at the Harvard Medical School and the Director of the CTS Signaling Laboratory and an immunologist here at the MGH. Today, webinar is titled Signal Transduction, Pathways to Transplant Tolerance and Rejection. And now I will turn to Sandro so he can give us the exciting lecture today. Thank you.
Thank you, Thiago. Exciting. I don't know. So before I came into the world of immunology, I actually was in the world of signaling. And I was in Ray Erickson's lab where we actually looked at the MAP kinase pathway, where I was involved in the cloning of MEK1. And I'll mention simply, you'll see it mentioned while I go forward. Now, the whole idea of signaling is basically to understand how a cell, in response to an extracellular signal, responds to that, translates that internally, and then responds to that signal in the appropriate way. Now, a lot of times, so before I go to my talk, I'm going to focus on four things. First, I'm going to give you a brief overview of signaling. Then I'm going to discuss some of the immune-related signaling pathways that we all work with, not all of them, but some of them. And then I'm only going to cover about the mode of actions of a few immunosuppressive drugs, two which target at the receptor level, one that targets a kinase specifically, and one that targets a phosphatase. And then I'll end with talking about one of my favorite kinases, glycogen synthase kinase 3 beta, which is a unique kinase. And we did some work 10 years ago, and I'll just show you some of the work we did with that. So signaling, as I said, is a way that the cell responds to an extracellular signal. Without a signaling pathway, you could almost consider a cell inert in response to its environment. So what happens during the presence of a signaling molecule, you get interaction with the appropriate receptor, and then a whole slew of events occurs, which involves phosphorylation as well as dephosphorylation. Phosphorylation is what triggers the activation of a pathway to an extracellular signal, a growth factor, viral DNA, bacteria, other interactions with other cells. And in turn, what happens is that you activate a molecule. Most of the time, it's a relay molecule. And these relay molecules are usually phosphorylated, connected, bind to the receptor. And in turn, upon being phosphorylated, they recruit kinases and other proteins to that complex, and in turn start activating downstream kinases. And downstream activations occur through either having kinases upstream, and then eventually reaching, as you can see here, an active protein. And now, when this occurs, it's usually a very quick response. And just as fast as it happens, you also get a dephosphorylation by phosphatases that remove that phosphate, inactivating the pathway, because obviously you do not want to have a pathway activated for a long period of time, because that leads to pathophysiologies. And a lot of times when there are mutants in any of these cascades, you get, as I said, disease like cancer or autoimmunity or other diseases because of a dysregulation of these pathways. Now, upon this active protein, it could either be a kinase or an actual transcriptional factor that moves into, moves into the nucleus, where you get transcription of the appropriate genes that get translated and therefore help in the response to that particular signal. Now, as I said, kinases are usually what govern this cascade event. And even though I won't mention phosphatases, I'll briefly describe what phosphatases do. So you have at least 3 known family of kinases. Tyrosine kinases, these are usually near the membrane. And these are the ones that usually phosphorylate a receptor upon the interaction of that receptor with a growth factor. And the phosphorylation on tyrosines usually is what helps, they act sort of like a docking proteins when you have a phosphorylated tyrosine, and proteins basically bind to those proteins, and in turn start recruiting other kinases. There are also dual specificity kinases, which I was involved with MEK1. And dual specificity kinases are interesting because they're both serine threonine kinases as well as tyrosine kinases. And they usually serve a role upstream of MAP kinases, and which includes the serine threonine kinases, ERKs, PKCs, and other AKT. These are all proteins that are serine threonine kinases. And the interesting thing about MEX1, they phosphorylate ERK on threonine and tyrosine. Without that dual phosphorylation, ERKs do not get activated. Now, similarly, once these proteins get phosphorylated and activated, you do not want them to be kept as phosphorylated proteins, because obviously they'll maintain activity and in turn could lead to, as I said before, disease. So tyrosine kinase, obviously there are tyrosine specific phosphatases. For dual specificity kinases, there are also specific phosphatases for that, as well as for the serine threonine kinases. So you have not only tyrosine, dual and threonine kinases, you also have tyrosine and serine threonine phosphatases. And upon the dephosphorylation of these, you get inactivation. And interestingly, while some phosphatases exist, getting ready to be activated to result in dephosphorylation, the MAP kinase pathway actually leads to the regulation, the transcriptional upregulation of its own phosphatases. In turn, they get transcriptionally activated, translated, and in turn come back and dephosphorylate and inactivate ERK. So there's various ways that actually pathways regulate themselves, but it's usually an interplay between phosphatases and kinases. Now, just because kinases exist, they don't phosphorylate any old protein. Each protein has to have what's known as a consensus site for phosphorylation. In other words, for example, ERK can only phosphorylate serine next to a proline, but you also have a proline, leucine, or proline X, serine, and proline. So kinases recognize specific sites on proteins and then phosphorylate those proteins. So not all proteins can be phosphorylated by all kinases. They have to have that specific sequence recognized by that particular kinase. And there are many ways actually, if you're going to study kinases, to go for. I'm just going to mention 3 ways in which we used to study the functionality of kinases. One way is obviously to mutate the phosphorylation sites. And you could either do a serine to aspartate or threonine to glutamate. Sometimes people mutate serine to glutamate, but just keep in mind that aspartate, the ionic rate AI for aspartate is similar to a phosphorylated serine. And again, the glutamate is similar to a phosphorylated threonine in size. For loss of function, what we usually do is mutate the serines of threonine to alanines and tyrosines to phenylalanine, in other words, basically mutating those. And again, basically what we usually do is take serines and make them into aspartates, and then you put those back into cells, and therefore it should mimic a phosphorylated protein of that particular protein. And that's how you actually study the functionality of what phosphorylation actually means, at least in cultured cells. In vivo, people have created now with CRISPR, you could actually mutate different amino acids and in turn regulate the functionality of these proteins. So there are various ways to actually study the function of phosphorylated protein by altering the phosphorylation site. One that was great, actually great for the signaling field was the creation of phosphospecific antibodies. These antibodies actually were created to recognize the phosphorylated forms of either kinases or transcriptional factors or other proteins that get phosphorylated. And what this allows you to do is actually follow these proteins, because they both work in flow, most of them work in flow cytometry, and a lot of them work in immunohistocompatibility, IHC. as well as immunofluorescence. And you could do a time course to see when these antibodies are phosphorylated, because again, when I talk about molecules, inhibitors, you want to know when those pathways are turned on, because you just don't want to add inhibitors for long periods of time. At least you want to target when those specific proteins get activated, and therefore target and add those inhibitors at that specific range of time. So I usually tell people that's what you always should do when you're going to try to use inhibitors. And in a way that also, because long-term use of these inhibitors can a lot of times be detrimental, and I'll talk about a little bit of the side effects that inhibitors could actually do. So as I showed you, phosphospecific antibodies, this is from our recent paper, and you can actually see that when you use phosphospecific antibodies, these are our toll structures, you actually see beautiful staining of tolls with phospho-ERK, which you don't see in a native kidney. Interestingly, and this is for some other time, you can actually see that usually phosphorylated ERK is nuclear. In the tolls, it's all membrane bound, which has a different functionality, which I could discuss in a future talk sometime. And as I said, we'll talk about small molecules, and in particular molecules that people use to actually study immune responses and regulate immune responses. And a lot of you using immunosuppressive drugs, as I said, use these kind of inhibitors, and I'll talk a little bit about 4 of them. So when we talk about signaling, what we're actually talking about, this is just one aspect. And I'm going to talk more about the TCR receptor and CD28, which both, when engaged by APCs, activate downstream signaling pathways. CD28, and this is not the only thing that they activate, but mainly when you engage CD28, you activate the AKT pathway, and the AKT pathway eventually leads to one of the things that it does, it leads to the activation of NF-kappa B, and NF-kappa B, as you know, is involved in a lot of inflammatory responses, as well as some anti-inflammatory responses. So it frees up NF-kappa B and REL to go into the nucleus and act as transcriptional factors. The activation of the TCR pathway eventually leads to the activation of the MAP kinase pathway, one of my favorite pathways, which in turn, ERK goes into the nucleus, phosphorylates Phos as well as Jun, and then the Phos-Jun complex, also known as AP1, also interacts with a transcriptional site as transcriptional factors and interact with promoters of specific genes. So these two together actually is a simpler way of doing. But when we're talking about signaling, and basically this is what's actually happening in the cell. All of these, and this is just part of what's happening in the cell and about engagement of all these pathways, you have this crosstalk between phosphatases, kinases, transcriptional factors, all happening at once, but in a beautifully regulated way. And when you talk about activation of these pathways, nature is done in such a way that in order to lead, for example, to a certain cytokine or a certain activation of certain genes, you require multiple pathways to converge. Without the convergence of these pathways, you don't get activation of that particular pathway, of that particular endpoint. So let's start talking about what happens when a T cell interacts with an APC. Again, simply looking at this, when you have the MHC presenting peptide interacting with the TCR and the CD28 interacting with the co-stimulatory molecules, you get activation of a T cell. It leads to proliferation and differentiation and effector function. And as I said, what actually happens inside a cell is this is partly what happens. And eventually what these transcriptional factors do is interact with specific promoters that either govern, in this case, let's just simply look at IL-2 and gamma interferon. When you see IL-2 and gamma interferon, the transcription of these molecules are dependent upon these transcriptional factors. And as you can see, you have the NF-kappa B, CREL, the AP1, which is Phos-Jun, NFET, which I'll talk about later, and Tbet, as well as STAF4. Now, a lot of these transcriptional factors are used cross genes. In other words, you could hear while they're pro-inflammatory cytokines, a lot of these transcriptional factors are also used in inducing the transcription of FOXP3 or PD1. And so nature is done in such a way where you have a lot of common transcriptional factors used across different genes, but then you have those unique factors that are specific to those cytokines. And in particular, in this case, when Tbet is on, you know that it's going to be involved if all the right transcriptional factors are there in the induction of gamma interferon as well as IL-2. So when you look at transcription, a lot of these transcriptional factors are used. You can almost say they're generic. Always look for those transcriptional factors that are specific, because those end up to be the better targets. Let's say if you want to talk about anti-inflammatory or pro-inflammatory responses. And I'll talk about Tbet later as we go forward. So this is, next I'm going to show you an experiment that you would do. Let's say you know that these pathways are turned on. You know that these MAP kinases turn on. You know that AKT is turned on. You know that these various factors that turn on. And something you could do is use these small molecules, which are not necessarily used in immunosuppression right now, but some of these molecules have been used in combating tumors. So, and this is a little experiment that I did a long time ago. This is with Gilles. And basically here looking at after skin allografts, doing an AliSpot and looking at activation of T cells in the presence of either direct or indirect pathway. And you can see that usually you get a nice induction of gamma interferon as well as IL-2, both in IL-2, both in a direct and indirect pathway. If you add NF-kappa B inhibitor, which would inhibit REL, one will predict that you get a reduction. And in fact, you do get a reduction in production of gamma interferon. And again, if you add inhibitors to MEK1, which would turn off this pathway, you should see a reduction as you do see here. And so basically this tells you that, that just inhibiting one pathway can bring the whole system down. And that's how people approach treating disease as well as using immunosuppressive drugs, because it's known that even though multiple pathways are needed to induce the transcription of these genes, all you have to do is knock out one pathway and you compromise the whole system. And similar, if you inhibit AKT, again, AKT also shows that it's important to inhibit gamma interferon as well as IL-2 transcription. So knowing this, this is basically how a lot of immunosuppressive drugs have been devised. And the first group that I'm going to talk to you about is one that specifically interacts with the costimulatory molecules, and that's balatacept as well as other CTLA-4 Ig proteins. And the idea behind these is that we know that the engagement of MHC with CD28 results in the induction of this pathway. Basically what CTLA-4 Ig does is basically interfere with this pathway. And in fact, and it's known that when you do only have T cells interact just with the MHC, eventually what happens is you get energy as well as apoptosis of T cells. And when you actually have a regular response, as I showed you before, you get activation. But if this, if CTLA-4 is present and interacts with CD80 and 86 costimulatory molecules, you prevent the activation of CD28, therefore leading to cell cycle arrest, as well as actually leading to sometimes cell death of these molecules. So you can actually see that just by interfering with one pathway, you also affect the outcome of the T cell response. And again, in that case, with respect to simply looking at CTLA-4, you probably compromise the NF-kappa B pathway, and in some extent the phosphorylation pathway, but mostly to simplify matters, you're basically getting rid of these transcriptional factors, which leads to the downregulation of IL-2 and gamma interferon and other genes. Looking at cyclosporine and tracholimus. Now, these two are basically the phosphatase inhibitors that I was talking about. A lot of people use calcineurin as well as FK506. And basically, this is how what happens during the activation of a calcium channel. When a T cell interacts with an APC, you also not only get activation of those pathways that I've showed you before, but you also get activation of a calcium channel, and the translocation of calcium from the extracellular to the intracellular modality actually binds to calmodulin. Calmodulin activates calcineurin. And what calcineurin does is take NFAT that's phosphorylated, this is an inactive form of the transcriptional factor, removes that phosphate, and then NFAT translocates into the nucleus, and again takes part in the activation of transcription activation of gamma interferon and other pro-inflammatory cytokines. Similarly, it also does affect the CREL and NF-kappa B, specifically also FK506, which is while cyclosporine tends to focus on this as much, the modification by FK506 not only affects this pathway, but for some reason, which still it's unknown how, also affects NF-kappa B. So that's why sometimes FK506, I think, may be a better suppressor, but a more potent suppressor, which you have to be careful about. And again, what happens is the NF-kappa B, as I said, you add the cyclosporine and tracholimus, and what happens, it inhibits calcineurin, and fat remains phosphorylated, doesn't go into the nucleus, and therefore what you basically do is compromise not only NF-kappa B, but MFAT, and therefore downregulating IL-2 and gamma interferon. But again, keep in mind, and I'll come back to this, a lot of these transcriptional factors are also important in FOXP3 or PD1 expression. And I'll cover that a little bit in the end, basically showing that when we use these inhibitors, keep in mind what other effects it might have, especially with anti-inflammatory cells. Now, this is a molecule that's being used a lot, Joran and everybody else using tocilizumab or the anti-IL-6 receptor inhibitor as well, siltuximab, which is the anti-IL-6. Now, I'm going to show you a picture of what interleukin-6 does, because it's an amazing molecule. And it looks complex here, but simply you have IL-6 receptor, this membrane-bound versus soluble receptor. And in both cases, what these receptors do is aggregate with GP130, and in turn, this aggregate, both through soluble receptor or non-soluble receptor, activates a whole series of pathways, the MAP kinase pathway, similar to what MHC does, as well as the JNK pathway, which is another, which also makes certain that certain AP1, FOS and Jun get also upregulated. So it's doing, in a way, maximizing the number of pathways that lead to the transcriptional factors needed for the activation of specific pro-inflammatory cytokines. It also has that JAK-STAT important step whereby JAK-STAT pathway gets turned on, where the STAT, as STAT3 or STAT4 go into the nucleus, activating again pro-inflammatory molecules. Even though later I'll show you that some STATs are also needed to induce anti-inflammatory molecules or suppressor molecules. And finally, it also activates the MTOR pathway. The MTOR pathway is a unique pathway, and I'll talk about this next. And it's the MTOR pathway that is actually needed in order, once you have all these transcripts being made, you need efficient translation of those transcripts, and that's what the MTOR pathway does. That's why a lot of people target the MTOR pathways in order to prevent, one of the things is to prevent the translation of proteins, of RNAs that could actually lead to the formation of cytokines. So so I'll skip this. And again, these inhibitors act on the IL-6 receptor as well the soluble IL-6, and in part doing this, what happens is actually you actually eliminate a whole slew of transcriptional factors. STAT4 usually comes from when interleukin-12 is involved. Interleukin-12 actually leads to this activation of STAT4, and interleukin-12 along with IL-6 usually co-express in an inflamed region. And as you can see, just by using, again, all these inhibitors, you're focusing on the alteration of transcripts that in turn can result in the downregulation of gamma interferon as well as IL-2 and other inflammatory molecules. But the interesting thing about, and this is a side note with IL-6, IL-6 is also under the right condition an anti-inflammatory cytokine, and it's known as a myokine. A myokine is usually, as I describe here, during exercise, what happens is secreted by skeletal muscles, and along with interleukin-1 receptor agonists and IL-10, all three together actually result in protection of the muscle and leading to stimulation of hypertrophic muscle, leading to muscle growth as well as myogenesis. So interleukin-6, in the absence of TNF-alpha or other inflammatory molecules, in the presence of the right anti-inflammatory molecules, can also be anti-inflammatory. And interestingly, certain tumors, such solid tumors, such ovarian tumors, when they secrete IL-6 and GM-CSF, and when they're secreted together, What these molecules do, they attract the immunosuppressive MDSCS cells, or myeloid-derived suppressor cells, to the tumor site. If you just have IL-6 or GM-CSF, the tumors are not protected. But when they're together, you actually create the almost like a chemokine for the MDSCS to come and protect the tumor. So when we're looking at interleukin-6, it's not just pro-inflammatory. Always keep in mind that depending on what other factors are there or other cytokines are there, cytokine functions may change from anti-inflammatory to pro-inflammatory, from pro-inflammatory to anti-inflammatory. So just something to keep in mind. So as I said, one of the pathways get activated by interleukin-6 is the mTOR pathway. And mTOR is not just activated by IL-6, other pathways activate mTOR. As you can see here, AKT, which is again activated through IL-6 also, as well as CD28. So when we talk about mTOR, we have to talk about rapamycin, and rapamycin is the known inhibitor for mTOR. And as I said before, not only IL-6, but this TCR-CD28 pathway can activate the mTOR pathway, as well as these pathways that lead to the transcriptional factors, activation of transcriptional factors. So while one pathway activates the transcriptional factor, as I said before, the activation of mTOR ensures that these transcriptional factors get translated, and then the cell responds to that particular pathway more efficiently, but that single more efficiently. But looking at mTOR, there are two types of mTORs. There's TORC1 and TORC2. TORC1 or mTOR in this region, what happens is that when it's activated, it leads to the increase of translation, as I mentioned before. And it's done two ways. You activate S6 kinase, which in turn phosphorylates ribosomal proteins to make certain they're more efficient in translating, but also turns off the anti-translation protein 4E-BP1. By turning that off, you ensure more efficient translation. And the way this gets activated requires the activation of at least the AKT pathway. And how does the AKT pathway work in regulation of mTOR? Well, that's where TORC2 comes into play, as well as other pathways. When you activate the AKT pathway, two things happen. You get the phosphorylation of AKT on threonine 308 by PDK1, which is a kinase that's dependent on PI3 phospholipids. And then you also get the activation of mTOR, which phosphorylates AKT on serine 473. So a lot of times when people look at AKT activation, they just look at one phosphorylation site. To actually show that it's fully activated, you have to use phosphospecific antibodies to show both are phosphorylated. And when both are phosphorylated, what AKT does is phosphorylate TSC1 and TSC2, which basically blocks Reb. Reb is a GTP binding protein that, when is blocked, mTORC1 remains inactive. But when you inhibit TSC1, freeing Reb, it activates this pathway. And also, a lot of times when people talk about low dose rapamycin and high dose rapamycin, what people have shown is that with low dose, you get phosphorylation of S6 kinase, but you don't get the inactivation for BP1. In other words, you don't get the phosphorylation and inactivation of 4E-BP1. When you have high dose, mTORC1 can phosphorylate both, and you get more efficient translation. So that's something to keep in mind when we're talking about low dose and high dose. At least this is what's known so far, how low dose and high dose rapamycin works. But of course, when you're looking at TOR, it's more complicated than this. It's more like this. And again, just looking at inflammation, and I basically went through it all, inflammation helps activate mTOR. In turn, you get the inactivation of TSC1, and in turn, you get activation of TORC. Rapamycin, in interacting with, usually interacts with a protein called FKB12, and this structure, this complex, is what inhibits mTOR. And interestingly, this is a molecule that I think some people, I think Jordan is interested in, DEPTOR. DEPTOR usually binds and helps inhibit mTOR. But upon activation of mTOR, DEPTOR gets phosphorylated and broken down. So by regulating DEPTOR, you can also regulate, along with rapamycin, MTOR function. So, as I said, when you look at transcriptional promoters and transcriptional sites, you want to look at what's common and what's not common between all genes. And as I said, one of the proteins that's really important in pro-inflammatory molecule, in the induction of pro-inflammatory molecules, is this transcriptional factor Tbet. Now, Tbet gets activated by various different kinases. It requires both STAT1 and STAT4 for its transcriptional activation. And again, STAT4 comes from when interleukin 12 is activated. And STAT1 is activated by various molecules, including the interaction between MHC and the TCR, gamma interferon receptor, type 1 interferon. You can read all this. So the activation of this leads to, as I mentioned before, not just the activation of gamma interferon and IL-12, but also activates other genes that can tend to be pro-inflammatory, as well as having similar functions in the activation of B cells in response to TLR activation, as well as gamma interferon activation. So one of the things that Tbet does, so remember, it's important for the activation of gamma interferon and IL-2, just to mention a few. But it's also inhibitory, and that's what some transcriptional factors do. So if you upregulate Tbet, it could go in the nucleus and turn off the PD-1 promoter. The PD-1 promoter, remember, is it's a protein that's expressed on the surface, and it's involved in suppressing an immune response. And a lot of times it's associated also with exhaustion. So the PD-1, PDL pathway is regulated by Tbet. But what's happening during an immune response? Because remember, an immune response is not chronic. It's only chronic under certain pathophysiological conditions. Usually you respond, and then you have to turn off that response. And the way that's done is, nature always does in a clever way, is that during an inflammatory response, you have upregulation of Tbet and other molecules that turn off molecules that you do not want to have expressed because they could turn off an immune response. And in this case, you have inhibition of FOXO1, which is important for the transcription of PD-1. But you also, at the same time, molecules that tend to be pro-inflammatory, like IL-6 and IL-12, their activation leads to the transcription of activation of STAT3 and STAT4 that binds to the distal regulatory element, excuse me, of PD-1 promoters. As these get upregulated, and as Tbet levels go down and FOXO level go up, as the AKT pathway gets downregulated after the initial stages of immune response, what happens little by little, you start getting activation of the PD-1 gene and the expression of PD-1 that in turn helps turn off an immune response. And so what actually happens once you have PD-1 expressed on the surface, PD-1 through the ITIM or ITSM sites on the protein help to turn on phosphatase called SHP2. This phosphatase turns off the AKT pathway, and in turn, by turning off this AKT pathway as well as also turning off the MAP kinase pathway, you could turn off NF-kappa B activation, IL-2 production, mTOR activation, as well as BCXL activation. So by little, the initial start of immune response is to be immunogenic. And as time goes on, you start expressing PD suppressor genes, that in turn help turn off the immune response. So it's a very intricate way of doing it, where you're actually seeing, at the same time you're seeing the activation of pro-inflammatory genes, little by little you start getting the same transcriptional factors that overlap, along with some unique transcriptional factors like FOXO1. Little by little, as the balance between immunogenicity and turning off the immune response is regulated, you start seeing more of the expression of these PD-1s. So nature has done in such a way that it's a beautiful balance between how to regulate a response, both either in the immune system or any other way. So when we're talking about, as I was telling you, a lot of transcriptional factors are used by pro-inflammatory molecules as well as anti-inflammatory molecules. And this is a good example is FOXP3. FOXP3 and expression, and something you have to keep in mind when you use these immunosuppressive drugs down the road. For example, you see here, again, to remind you, these are some of the transcriptional factors that are used by IL-2 and gamma interferon, with the unique one being Tbet. And remember, STAT4 is also used for the induction of PD-1 as well as FOXJUN, FOXO1. When you look at FOXP3 expression, FOXP3 expression is very well regulated. You have 3 conserved non-coding sequences, 1, 2, and 3. 2 is where usually gets turned on when methylated and leads to the production of thymic or regulatory Tregs. When you get methylation of the CNS1 section, you favor more the induction of the peripheral, also known as iTregs or induced Tregs. And what's conserved between the, what's common between the two is CNS3, which is between exon 1 and 2 of FOXP3, and REL binds to CNS3, as well as you have all these other proteins that bind to the regular promoter, along with these other unique proteins, leads to the induction of FOXP3, as well as the phenotype of the specific regulatory cell. In other words, thymic versus induced. And as you can see here, remember, NFAT is also needed for IL-2 and gamma interferon, as is CREL, as is AP1 FOSJUN. So you have, again, these overlapping transcriptional factors. But what's unique with FOXP3 is that you also, like in PD-1, you need FOXO, FOXO1, SMAD3, as well as RUNX and all these other genes, as well as FOXP3 coming back after being translated, binding to the promoter to continue the maintenance of FOXP3 expression. And more specifically, I'll show you in more detail what's actually happening. So when when we induce regulatory T cells in vitro, you basically at IL-2 and TGF-beta, these two alone basically don't have very minimal effect in the induction of regulatory T cells. But when you have them together, you get massive induction. And why is that? Well, as I said, SMAD3 is important for the induction, and SMAD3 comes from the induction of the TGF-beta receptor family. IL-2 is important for allowing for STAT5, the JAK-STAT pathway, where STAT5 gets regulated and goes in and helps with the regulation. And remember, MTOR inhibits FOXO1. It also does the same thing for PD-1. So inhibition of MTOR, and that's why when you have rapamycin, you get better induction of regulatory T cells, is because you're turning off MTOR, which allows for the expression of FOXO1, contributing to the induction of FOXP3. So you have, again, you have a lot of transcriptional factors that are common between pro and anti-inflammatory molecules, but depending on the specific genes like RUX1, ET1, SMAD3, these are the genes that are really important. And then to maintain FOXP3 after being induced, you get the transcription of FOXP3, gets translated and comes back, interacts with the appropriate transcriptional factors, maintaining the expression of FOXP3. So in the last part of my talk, I'm just going to focus on GSK3 beta, or glycogen S6 kinase. And why am I going to talk about GSK3 beta? GSK3 beta is the only kinase, known kinase in our body that is always on. Other kinases have to be turned on through activation by growth factors, but GSK3 is always on, and its main role is to make certain that certain genes like beta-catenin are phosphorylated and degraded, because when beta-catenin is upregulated, you get activation of growth. And a lot of tumors have it in such a way where you actually lose the regulation of beta-catenin. What happens, you actually enhance tumor growth. So right now in our body, GSK3 is on in all our cells. So one of the things you have to, upon an immune response or any growth factor, you want to make certain that you respond efficiently. And one of the kinases that gets turned off is GSK3. And you have to turn it off, because otherwise you can't respond efficiently to growth factor or any cytokines or things like that. And basically, as to recall, remember the MAC pathway downregulates GSK3, as well as the AKT pathway negatively regulates GSK3. And both of these pathways phosphorylate GSK3 on serine 9. Remember, the MK pathway can be turned on by the MHC TCR activation the AKT pathway, one of the ways that it gets turned on by the costimulatory molecules interfering in binding to CD28. So these pathways went on, turn off GSK3, allowing for growth and appropriate response to growth factors. And the reason why you want to do that, besides regulating beta-catenin, GSK3 has also been shown to phosphorylate Tbet, inactivating Tbet, and therefore IL-2 and IgA interferon levels go down. It could also phosphorylate and activate another MAP kinase called JUNK, or Janus kinase. And by phosphorylating and activating JUNK, it could also downregulate IL-2 and gamma interferon. So it makes sense that pro-inflammatory responses at least need to turn off GSK3. And also, remember about when you activate the calcium channels. Calcineurin gets activated, dephosphorylates and fat goes into the nucleus. But if this was maintained in a chronic state, what would happen? You would have, for example, pathophysiologies occurring. So one of the roles that GSK3 does after this pathway is activated is GSK3, activated GSK3 can actually go into the nucleus, phosphorylate and fat, and then a phosphorylated fat then basically goes back into the cytosol, inactivating that molecule. So it has a lot of inactive properties. And so one of the things that we also did just to play around with this molecule, if GSK3 regulates IL-2 and IL-6, gamma interferon, then inhibition of GSK3, again, in an ELSpot analysis assay that we did using skin grafts and memory, one would expect an upregulation. And in fact, what happens by using inhibitors like small molecules that specifically inhibit GSK3 is that, in fact, unlike NF-kappa B and MEK1, which inhibit the response, when you inhibit GSK3, you get upregulation of gamma interferon and upregulation of IL-2. So suggesting that, in fact, it makes more sense that when you inhibit that, you enhance the pro-inflammatory response. And this you could also see in a dose-dependent manner. I'll skip this. But another interesting thing about GSK3, and this at the same time that we were doing these studies with GSK3, a paper came out by Ding et al. That basically showed that beta-catenin, our stable beta-catenin, can in turn, maximizing expression of beta-catenin results in Treg cell stabilization and enhanced survival in vivo. And this is a little paper that we did 10 years ago. I'll skip this. Basically looking at the fact, what role does GSK3 play in the regulation and function of FOXP3? So as you recall, CD28, activation of CD28, again, this is very simplified. You have activation of AKT. In turn, AKT phosphorylates GSK3 on serine, inactivating it. But as I said, when you don't have serine phosphorylated, GSK3 is always on, and what keeps it on is phosphorylation on a tyrosine group. So when you have just phosphotyrosine, GSK3 is active, and beta-catenin is degraded. When you have inhibition of GSK3, the serine is also present, and it turns GSK3 is inactive, allowing for beta-catenin to be stabilized and go and transcribe. So we did an initial analysis of naive CD4, CD25 high T cells. And as you can see here, and this was published in 2010, you can see that CD25 high molecules also have high levels of phosphotyrosine, suggesting these are very active. This is very active GSK3. And to make certain that it's active, we also use phosphospecific antibodies to beta-catenin that recognize phosphorylation on serine 33, 37, 41. These are the sites that are phosphorylated by GSK3 inactivating GSK3. So you see where you have high levels of phosphotyrosine, which is the high CD25. You also have this bend, which shows also a high level of phosphorylation of beta-catenin. So in the naive CD4, CD25 high levels, CD25 high cells also have phosphorylated and probably destabilized beta-catenin. And when you look at FOXP3 staining is also with those high CD25. So FOXP3 positive cells are also have a very active GSK3 and somewhat destabilized beta-catenin. But basically, then we want to ask, well, according to the Ding paper, if you stabilize beta-catenin, you get stable Tregs. So we asked the question, what happens if we have GSK3 inhibitor to a suppression assay? So we looked at this and basically saw that when you have T cells alone, CD25 negative, CD4 positive cells, with CD3, CD28 microbeads, you could see that you get nice induction, as you can see by thymidine uptake. When you add inhibitor, you get further induction, and it's been shown that with GSK3 beta inhibitor, and remember, you're stabilizing beta-catenin, allowing for growth to increase, that you actually see an increase in proliferation, which is good because it shows that when we add inhibitor and increasing amount of Tregs, you get more efficient suppression when you have the inhibitor, as you can see here, versus when you don't have the inhibitor. And in fact, the suppression is even higher because you're going from a higher level to a low level. As you can see, 86 versus 65, and eventually you have 95% inhibition. And when we looked at BCXL, which is an anti-apoptotic protein, through flow in the paper, we showed that that's stabilized in FOXP3 cells. But we also showed that BCL2 in isolated Tregs, 42, 40, and 66 hours incubated with inhibitor or without, you see that BCL2 expression remains higher in the Tregs that have the inhibitor, suggesting that's the reason why we're actually seeing better suppression because of more stability of the Tregs. And we did this simple experiment that's also was in the paper. This is work done with Kang Mi and Jim Markman. And basically when we injected 100 micromolar just of the inhibitor, which is not the best inhibitor. Since then, more soluble inhibitors have been found. Injecting this daily, you actually get better outcome in islet survival. And this is just by injecting this inhibitor. And one day when either we go back and revisit, it'd be interesting to see better inhibitors that are more soluble, like bio, which exists, to sort of see by injecting bio in this similar experiment, do we actually see a better protection. So basically, GSK3 seems to be both a pro as well as an anti-inflammatory GSK kinase. It's always on, and one of the things that it does, inhibition of GSK3 helps potentiate Treg suppression by stabilizing the Tregs, and that is done by upregulating and maintaining BCL2 as well as BCLXA. So one of the things that I always do, looking at FOXP3, and besides stabilizing beta-catenin, are there sites in on FOXP3 there are GSK3 consensus phosphorylation sites? And in fact, they are, and they're conserved in mouse, human, bulls, and macaques. In future experiments would be that, what if you modify this protein? Do you stabilize FOXP3 even more? And therefore making a more efficient suppressor. But that's something for the future. But this is the last slide, and most recently, this is the most recent JX Med paper. People found the stabilization of beta-catenin also induces the transcriptional expression of PD-L1 in glioblastoma. So you can see that the stabilization of beta-catenin not only helps stabilize FOXP3 and FOXP3 expression in Tregs, but it supposedly also has a function of maybe increasing the expression of PD-L1. So with that, hopefully I didn't lose a lot of people. Thank you, and I'm happy to take any questions.